Okay, so we are going to make some recombinant DNA, and we have our list of things we need to put in order. So we can even flip back through and look at the slides. But the very first thing we needed to do was to start by getting the DNA from the humans and making tons of copies of it. So we used PCR to do that. PCR made billions of copies. Now, we had to do something to those billions of copies of DNA to be able to put them in with the plasmid. So we had to cut them with restriction enzymes to give them sticky ends. Once we had them both cut, we could glue them together using DNA ligase. And now that they're glued back into a nice pretty circle, we can introduce the DNA into bacterial cells, and then we can plate those cells onto medium containing antibiotics. So I believe that is answer C down here. So that gives us a way to then grow up lots of bacteria that have our plasmid of interest without us needing to do very much work. Literally, this is like everything that y'all have done in class. Like, you could do this. I guess we haven't done the PCR directly. We would have been doing that this week in class. I guess technically last week, but storm. So researchers did sound tests on normal and FoxP2 transgenic mice. What's the most reasonable conclusion drawn from the figures? So down here, we could see that normal mice are shown in white, transgenic mice are shown in black. PF stands for peak frequency, so of their squeaks. And we have the start PF, the mean PF, that's the average, the maximum, and then the duration. So is it the sound duration of the transgenic mice is less than the mean peak frequency of normal mice? Transgenetic mice have a lower mean peak frequency than normal mice. The maximum peak frequency of transgenic mice is similar to the mean peak frequency of normal mice, or D, all of the above. So what is the most reasonable conclusion that you can come from? The most scientifically sound conclusion? You should get B, because what you don't want to do is start picking stuff out of one graph and comparing it to another when it's comparing two totally different things. It's an apples and oranges situation here, that duration should not be getting compared to your peak frequencies. Apples versus oranges here. You want to compare within the same category. So B says that transgenic mice have a lower mean peak frequency than normal mice. Yep, that is true. You don't want to compare one's average to the other's maximum. That's not fair to do. So again, apples versus oranges, but even within the apples, make sure you're comparing Granny Smiths to Granny Smiths and not to giant red apples, whatever. So I also want to point out these little asterisks here. If you remember back when we did our chi-square analysis and you had p-values to find out if something was statistically significant or was it random chance, one asterisk means yes, it was statistically significant. Two asterisks means it was very significant. Three asterisks is super duper significant. And usually in the caption, it would tell you what their cutoffs were. It's usually like 0.05 and 0.01 and then 0.001 or something like that. But they will always define what these asterisks mean in the caption. So as in the next few weeks, you're going to start trying to find your own journal articles, and those are the kinds of things that you're going to be paying attention to. So some applications of transgenic organisms, like Alzheimer's pigs. So sometimes they will create an animal model, like a transgenic pig that has Alzheimer's disease, because they can do medical research on it easier than they can do on humans. You don't have the risk of, well, one, having to wait until somebody has the disease to start working on it. You could start working on preventative measures. You don't have as long of a lifespan on a pig versus a human, so you can do much faster research. There's less ethical concern with testing the brain of a pig versus taking the brain out of a human. Some people tend to frown on that a little bit. Another example here is BT cotton. So BT is a gene that they found that produces a chemical that crystallizes inside insects. So they used to just put this bacteria straight onto plants, and then as the pests ate the bacteria when they were eating the plant, they were getting the toxin. And they went, wait, we could skip that step and just put the gene for the toxin straight into the plant. And that's what they did. So now if anything that has BT in it, like corn or cotton, if an insect tries to eat it, they will die. And it can't hurt humans or any other mammals because the toxin only crystallizes in the conditions that exist inside the insect. It won't cause any harm to a mammal trying to eat it. So that's how it is safe to eat GMO foods. But again, these are just examples of why we would want to move genes around. So the patent below is from the USPTO patent applications. You're interested in investing in it, but your partners are concerned about food safety issues regarding the milk of transgenic animals. So this is for the process for producing exogenous protein in the milk of transgenic mammals and a process for purifying proteins therefrom. So breaking that down, exogenous, exo means coming from an outside source. There's exo and endo. So when something's endogenous, it's produced within your own body. Exogenous means it comes from an outside source. So we're producing outside source proteins in the milk of a mammal and then being able to purify those proteins out. So you can come back and read this if you want, but what they're doing is putting the gene for human insulin into the milk. And it explains it. They can't just put the gene into the animal and just say, ta-da, human insulin, because the animal creating a bunch of insulin inside it is going to kill the animal. It has a normal amount of insulin that needs to stay there. So what they've done is that they have put in a modified insulin precursor. So it's not mature insulin yet. And they make it so that it is under the control of a beta-casein promoter. That's going to make it only get produced in the milk of an expression vector, that's plasmid. They then transfect the expression plasmid into fetal bovine somatic cells, so baby cow body cells, such as fibroblasts, and enucleating the bovine oocytes by doing nuclear transfer. So they went in and took the nucleus out of an egg and put in the one that was modified, and then transferred those eggs into the pregnant mama cow. Mama cow then gives birth, and once baby cow grows up, baby cow's milk is going to be making the human insulin precursor, which can then be converted into human insulin. So, simply put, human insulin, growth hormone, DNA, is introduced into mammals such as cows. The insulin DNA results in the production of insulin protein, which is secreted in the milk of transgenic cows. The milk can be collected and purified as a biopharmaceutical product. Suppose that the transgenic cow or transgenic milk does not contain cow cells. Now, working groups write down where 1 through 5 would eventually be and use arrows to connect the movement of 1 through 5 among A and F. So I'm going to give you a little bit of time to think about it before I give the answer. So we have to think back to when we were doing our PCR and stuff from before and what steps have to occur. So this is combining the last two things you did with the mouse cells and what you were doing with the PCR and restriction enzymes and stuff. So I'm gonna try to draw this out for y'all on a white screen, and it's gonna be terrible. I apologize. Okay, so we can make it a little smaller. We want to start off, we need to have a human. Here is our human. Pink. Yay. So, this is humans have human DNA. We then want to take out that human DNA, and we want to extract human DNA and copy DNA, specifically the insulin gene, using PCR. We are then going to go to that next step, which was to do, we're going to cut with restriction enzymes. Ligate into a plasmid. And now we want to jump. I guess I can. Put the plasmid into bacterial cells. So we're going to transform into bacteria. That's going to then duplicate, extract our plasmid. We can now put the extra copies into transfect into cow egg. Now, they did mention that they were specifically going to put it into somatic cells and then put the somatic cells in, move the nucleus from the somatic cells into the zygote, but that's, we can, so we'll just say transfect into cow zygote. That goes into a prego cow or pseudo prego cow. All right. Baby cow is born with the transgene in all cells of its body. Now, baby cow grows up. baby cow grows up to produce the milk. All right. So looking back, making sure human insulin DNA, human insulin proteins, plasmid DNA, cow DNA, bacterial chromosome DNA. Okay. So just a reminder that like the bacterial cell is still going to have its own bacterial DNA. You don't take out the chromosomal DNA. It keeps its bacterial DNA. And you know that just like in class when we extracted the plasmid, we made it to where the chromosomal DNA was really heavy and went down to the bottom of the tube, where the plasmid tube was on the liquid. That was what you transferred the liquid over to the filter. And when you put that into the cow still has all of its DNA. You're not taking out the cow's insulin DNA or anything like that. It still has its own DNA. It's just getting extra DNA. And when the milk is made, now the milk is going to contain, color here. It's going to contain the human insulin protein. So the next part here is it asks you, which of the following would be in Mike's body after he drinks the milk of transgenic cows? Suppose that the transgenic milk does not contain cow cells. It's only going to contain what was excreted out of the cells. So, is Mike going to get human insulin DNA? He should have his own, honestly, but he's not getting it from the cow's milk. That DNA would stay inside the cow cells. Only what was excreted, which is the human insulin proteins, would be going into the milk. The plasmid DNA is what contains the human insulin DNA, and that again should stay inside the cow cells. The cow's DNA should stay inside its cells, and the bacterial chromosomal DNA should have never gotten transferred to the cow in the first place. It should have only transferred over the plasmid DNA, which contained the human insulin DNA. So only the human insulin proteins should be going into that milk. So normally I would have you discuss whether or not you would want to invest in this patent, thinking about the potential impact on cows, milk, and human beings, and listing out the pros and cons. And yes or no for would you invest in the patent. This opens up the kind of the conversation about animal life values versus human life. And I would say that it would be much better to take the insulin from the milk, because we know it doesn't harm the cow to make milk, compared to the ways that we used to have to get insulin, which would have involved taking it from animal sources that were more invasive, either completely killing the animal or just taking it from the blood. And that was much more troublesome than just taking it out of milk. So that would be the hope on why we would use this type of technology, that it would be a lot better on the animal. If we could get it from bacterial sources, it would be best. So there's credits for you. Okay. Anything about this that y'all want to talk about, or anything before I jump over to wrapping up for review stuff to get ready for exam 1?